my distinct pleasure today to introduce our speaker, Dr. Isabelle Laforest-Lapointe. She is the Early Career Canada Research Chair in Applied Microbial Ecology, so this is a prestigious award that she's received and I believe will be looking to renew soon. Dr. Laforest-Lapointe got her Bachelor's of Arts at the University of Laval and then went on to do 2 masters in Spain, one in statistics and one in terrestrial ecology and biodiversity. From there, she moved back to Canada and got a PhD in biological sciences at the University of Quebec at Montreal, with a mention of excellence for her work there. And this is really where I became familiar with Isabelle's research. She did a lot of work on tree-associated phylosphere microbiology, had a big publication in 2017, where her work really showed that actually we need to be thinking more about phylosphere microbial communities and how they influence the greater plant community ecosystem and function. And I think before that, many people were really thinking more so about the rhizosphere, let's say, than thinking that the phylosphere is something to really focus on. And then from there, she went to the University of Calgary, and that's where she started to do some work in animal systems. And as Isabelle was pointing out yesterday, that kind of your career trajectory kind of encompasses sort of a One Health aspect to it. So I think, she has a lot of great stuff to share with us today. And then for all of the speakers that come, we have these plaques that we've been giving out to say thank you. So.
Thank you very much for taking time in your busy schedule. So my name is Isabelle. I'm a Francophone, so I'll do my best to speak to you in English. But if I make mistakes or I don't pronounce well, now you know why. And this is my first plaque, so I'm really happy. Thank you very much for this attention. So it's a privilege to be here today. I became a professor in 2020, January, so you can imagine what it has been like in the last few years. And I've gotten a few invitations, but this is probably one of the few that really looks like an interview, very busy schedule, but it's also such a privilege to go back to this habit of being able to meet other scholars across the world. So I really appreciate the invitation. So the first thing I want to do today is feedback a little bit on Kevin's presentation. And this is intended for graduate students, because I think everybody here that became a professor has a different EV or a different course of story of what they decided to do, where, when, how. So I wanted to talk a little bit more about my career and early career, let's say. And when I started my BSc, actually, I didn't know what to choose between biology and maths. So I really didn't know what to do. And I just chose biology because I thought there would be some math in it and not the reverse. So I ended up doing my BSc in biology, and I had the chance to do a semester abroad in Barcelona. And you can imagine how wonderful it was. Nice wine, nice sun. It was amazing. And after coming back and finishing my BSc, I wanted to continue living in Barcelona. And I had I have two parents that are academics. So it was easier for me to justify to my parents that I wanted to be in Barcelona to study than just to be there and backpack and enjoy life. So I went and did a first master in ecology and really enjoyed my experience. But what I discovered was that I really liked R and I really liked statistics and programming. So my supervisor at the time told me, well, you should maybe go get some more education in this field. And there's this master of statistics at the Polytechnic school downtown in Barcelona. By the way, it's in Catalan. So Catalan is near Spanish and French. It was my fourth language, and I decided, yes, I can do it. It was the most difficult year of my life. I didn't get good grades. I was just scraping by, but I learned so much, and it's probably what made me so good at research right now, because I'm not scared of stats anymore. And I think that just losing the fear of seeing a statistical test and not being like, ooh, this is what is this thing, it really helps to just look a little bit more into it and try to apply it and master it. And so I came back and I wrote to 2 professors to do a PhD. One was with 3 functional traits, and that fed back on my ecologist side, and the other one was in DNA. And I said, I don't want to work with DNA. I'm not going there. So I went with the tree functional traits person, but that person was working on tree microbial ecology. So I actually worked for my PhD on DNA. So if you think you're choosing in your life, you're actually not choosing. Life is choosing for you. But I love my PhD. It was really fun. I looked at tree microbe interactions. I'm going to present the work today. And finishing my PhD, my supervisor said, you need to know who you are in a few words as a scientist. What defines you? And it was difficult to think about this because you need to be able to have a perspective on something that you're still doing, a growth that is still occurring. But what resonated with me was that I was a microbial ecologist. I was still an ecologist at heart, but what I loved the most was understanding how microbial community assemble, how do they evolve, how do they interact together. But I knew I could apply this across other fields than just plant microbe interactions. And I wanted to become a professor. It was something that I wanted to be. So I strategized and decided to go on the human side because I knew it would be better funded and it would give me an edge when being recruited if I could display skills across a wide array of fields. So I went to Calgary in the human health department, medicine department, and I learned about the immune system. It was again challenging. So you can see a pattern. I keep going out of my comfort zone. It's not easy, but I learned a lot doing it. And I managed mice cohort and I sacrificed mice and I fainted twice. So I decided that my sacrifice wasn't for me, but I did it and I learned a lot from it. And I finished my postdoc and I got an offer to become a professor at Sherbrooke, which was a town where I had never been. But I really loved the feeling of the department and the support that it provided. And so I went there, and after 1 year of mat leave, I became a professor in January 2020. So this is my resume. At the end of the presentation, you can ask me questions about it, or afterwards, I know I'll have lunch with the students. So just feel free to ask me questions. It's going to be 4 years that I started this January, so I'm pretty early career, but I've been having a lot of fun. And I would say that having a department that supports you is really the key to having a positive life in academia. Okay, so microbial ecology, what is it? And I feel that here you have the One Health Microbiome Center. You might be doing some ecology and you don't even know it. So microbial ecology for me is really trying to understand the dynamics of complex communities that include organisms that we don't necessarily see. So this can be only bacteria if this is the sole focus of your research, but it can also encompass micro-microbe interactions. You can be interested in who you find in a specific niche. It can be only the soil, it can be the gradient of a whole plant, it can be the human gut. But often you're asking very similar questions. You're interested into eco-evolutionary dynamics of a community that evolves through time, that interacts between kingdoms, not only bacteria, but fungi and phages, and even protists. And so this has been my cornerstone. This has been my go-to subject that I can take into any system and try to study. So this is going to be the core of my presentation. And when I started to interview, which many of you will do in the next few years, you need one slide that shows the faculty in the department who you are as a scientist. What is your professional card? What do you do? And so this is the slide I built when I started to interview. So I am interested to study microbes. Microbes include bacteria, fungi, microeukaryotes, and viruses. I'm starting to do some work on the virus now, but you will see from my presentation that I'm already way overextended. So I'm trying to bring it back a little bit. But I'm still interested into what is their role. You cannot put blindfolds and say they don't exist or they don't affect my communities. They do. And the holes that I'm interested into go from model organisms such as Arabidopsis thaliana to trees, which are long-lived organisms that are way more complex to study than just Arabidopsis, but also mice and humans. So I really like that I have been bridging both human and plants. And the skills that I can provide to the table go from culture qPCR sequencing short marker and long sequences, metagenomics to see not only who's present, but also what are they doing, even if our tax or functional annotation databases are still very limited. And finally, metabolomics to not only look at who's present, what we think they're doing, but also what are they producing in terms of molecules that can directly interact with cells from the host. And this is my framework. So I'm really interested into answering the question in host-microbe interactions, who runs the show? Is it the host that is able to select and manipulate its host, its microbial community? Is it the microbes that really influence their host and its fitness and then are the ones driving the show? Is the interaction between microbes what really matters? Or is it just the environment that selects and puts pressure on the host and then on the microbes as well? So I'm trying to see my career around answering these kind of questions, and my different projects will be somewhere alongside this continuum. When I was doing my postdoc in the human field, I still was thinking about what were the questions that were yet to be answered. And when you start a postdoc, you have to produce, you have to publish something, and data sometimes takes time to come to you. So I was trying to reflect on my field, my expertise, and find and produce pieces that would be perspective reviews that could put something on my CV, but at the same time didn't really need the data. And one thing that we did with Brianna Whitaker is we thought about what were the limits of the field of the phylosphere microbiome studies. But this really could apply to any microbiome at the time. We didn't really understand yet what were the temporal and the spatial variation or dynamics in microbial communities. At what scale should we study microbes? Is it in terms of hours? Is it in terms of days? Should we study a single leaf out of a whole tree? Or is it more a bunch of leaves or many trees? So the question of the scale still really matters. Metagenomics, I think, is still a limit. We are trying to completely assemble genomes of these microbes that we cannot culture, know what function does these microbe have. Even for organisms such as E. coli, even half of the genome still, we don't really know what's happening with these functions, these genes. So it's still a limit. And one thing I wanted to do is not only do bioinformatics. I wanted to culture. I wanted to have my own collections of microbes so that I would not always be just not seeing what I'm working with, but also manipulating organism. This can be daunting as a task because it's not easy, but I think it's really valuable, especially when you work in environments that are not the usual, environments that are less studied, that are very rich. To have your own collection of microbes to play with is very, very rich. And finally, trying to look at microbe-microbe interaction. And I think this is, as an ecologist, was something that I wanted to push forward. So I'm going to structure the presentation of today in 2 sections with a bonus, so a short third one. So the first one is going to be on plant, the second one on human. So the first one on plant microbes. So we know that plant and microbes interact, and most of the work had been occurring, like Kevin said, in the rhizosphere, because we knew it was so important for access to water, nutrients, and interactions with fungi, which in the world of the plant microbe interaction are really a key driver of the capacity of plants to grow. But it's not only fungi and it's not only rhizosphere. We've learned in the last few years that there are microbes on the leaves that can also influence the host growth, not only as pathogens, but also as symbionts. And that goes beyond bacteria and fungi. Some protists are present, they can predate on the bacteria on the leaf, and viruses definitely play a role. And even the role of the sun, the light, can have an impact on plant microbe interactions, which is what we recently published with Gaëlle Lajeunesse in Nature Communication, how having access to light or constant light can influence your resistance to pathogens. So when I started my PhD, there was this review on phylosphere by Julia Verholt in Switzerland. And this really launched me into trying to harness my PhD around the phylosphere. But beyond just pathogens, going into what are the positive interactions or the benefits of the microbes on the leaves of plants. And in the last few years, we've discovered that not only do microbes have a role on the host, but at a higher scale on the ecosystem. How do they do that? They can influence plant productivity, resistance to pathogens, and resilience to abiotic stresses like drought. And in the grand scheme of things, this means that plant microbe interactions at the host level also have a bigger impact at the community level, so at the forest ecosystem level. And everybody talks a lot about it everywhere in the world right now. So this is big when we think about global change, especially for plant organisms that are sessile. They cannot escape difficult conditions, at least not quickly enough to adapt to the changing conditions. So many researchers in forestry have been wondering what's going to happen with forest ecosystem or forest species. Are they going to crash? Are they going to go extinct? Are they going to move forward like the trees in The Lord of the Rings? Are they going to just compress and have a smaller realized niche because of the new conditions? So this is something that really got me interested into trying to see how plant microbe interactions could help us predict what was going to happen with species distribution. In forestry, we think often about the impact of hotter drought, fires, pests. We rarely incorporate microbes or just beneficial microbes in for capacity to forecast the sustainability or the resilience of an ecosystem. Okay, so the plant microbiota, it's been where I've done 75% of my work, and it's still what I do most of the time as a researcher. I've asked many questions, and today I'm going to present a few of these projects. Some I'm not going to cover, the one on moss microbe interactions and the one on soil virome I'm not going to cover, but I'm going to present to you 5 questions. What is the influence of the host? How do plant microbe interaction influence community productivity? What happens in the urban environment? And that might resonate with some of you for the One Health question. And what is the impact of species distribution, plant microbe interactions and species distribution range? I said 5, but I'm actually going to do 4 because I wanted to have some time to talk some more. And I'm going to focus more on the phylosphere because this is what my PhD was about. It was about plant microbe interactions on the phylosphere. Not that doesn't matter to study the rhizosphere, just because I needed an edge for my PhD. I needed to do something new, and we chose the phylosphere. And the phylosphere looks like this. It's kind of this lunar environment that you might think is really inhospitable for microbes, but it still harbors a very high diversity of microorganisms that are expected to be able to live in an oligotrophic habitat. It's just a fancy word to say it's poor in nutrients. And they can enter the apoplast on leaves to use the resources or the exudates of the leaf, carbon exudates or other metabolites, and they can influence the plant humidity either by being on the surface or coming into the apoplast and producing themselves effector proteins, for example. So first project of my PhD, I wanted to know if I look at a sugar maple and a red maple and a balsam fir and a paper birch, which are all tree species, just to be clear, I look at them and they're next to one another, will they have the same bacteria on their leaves, or will they differ in terms of community composition? And I wanted to know if I go and sample early in the season and later in the season, will I find the same microbes? And finally, if I cover the whole range of the temperate forest in Quebec, will I see similarities, or will it matter? Will the geographic location matter? So I set up, so this is where my university is right now. This is Montreal. You can see the square where this is. And we right now are somewhere here, if my geography is not bad.
Close enough.
Yeah, thank you. And this is Vermont and Maine. Everybody's situated. This is like 9 hours of drive. So I decided that for my first summer of PhD student, I would drive around Quebec like a mad woman to get all my samples. And I would sample 5 tree species at 4 different sites of the temperate forest, the 4 red dots, across 3 months, June, July, and August. And what I would look at was who was present on the leaves of the trees. What you see here is an ordination. And when I mentor my graduate student, I tell them, if you show a figure, make sure that your audience understands it. So this figure, it's not a statistical test. It's just a visual tool to see how communities differ in terms of composition. These communities, they have sometimes hundreds of bacterial taxa. If you try to see them on a 2-axis graph, it's going to be difficult. If you try to see it on a 100-dimension graph, our brain just cannot compute. So we use statistical techniques to reduce this multidimensional distance on 2 axes. This helps us synthesize the difference between the communities. So each of these dots, they represent 1 tree, 1 sample, 1 community composition. If the dots are close to one another, it's akin to being close by in terms of composition. The composition of the bacterial community is similar. It's not linear. It means that if there's 1 centimeter between these 2 dots and 1 centimeter between these 2 dots, it's not proportional. So it's an index of similarity. So, and the ellipses here represent the 95% confidence interval of each of my different tree species. So what you see here are the 2 sister species of maples that assembled communities that looked alike, but they differ a lot from the conifer species. And conifer species are tree species that keep their needles in the winter. So they don't shed their needles, they keep them. They probably accumulate different type of bacteria because of that. And they also have very different cuticles than the deciduous trees. It's much thicker in terms of waxy layer, and it has also a different component of a different concentration of nitrogen. Sorry. So you have the 2 conifer species, the 2 maple species, and finally the paper birch, which was like a complete outlier across my 5 species. If you are in forestry, well, you know that paper birch actually likes sun. It hates shade. So it always exposes its leaf in the sun. It will go at the top of the canopy and it will have its leaf there. So what is it exposed to? Sun, UV rays, I think is the right word. And also dispersal by wind and rain. So it was just exposed to a different set of possibility to be colonized. Probably also has different properties of the leaf. So I call paper birch my lady in the sun. It just liked to be exposed, and because of that, it assembled a different type of bacterial community. But what was fun is that this is not an experiment. Did I control for all the factors? No, I didn't. I didn't control for temperature, precipitation, history of management in the forest. I didn't control for many other factors. And still, with a very simple model, we explain more than 60% of the variation in community composition. Just host species explain more than a quarter of the variance in assembly of the community. So it seemed that host species identity really mattered in terms of who is present on the leaves of trees. And this is interesting because these trees were thousands of kilometers apart. And sugar maple assembled very similar community, even if it was thousands of kilometers apart. So very intriguing as a first project. But what's the point of studying these communities if it doesn't relate to the productivity of the tree or the function of the ecosystem? So I had the chance of working with a real experiment where we controlled a few of these factors, in which we decided which plants would be present in our design. And so this is a fully replicated factorial design where you have 4 blocks that are replicated. So you have 4 times the same experiment in different spatial organization. You have 2 gradients, I'm going to talk about just after this slide, and 19 tree species involved in the project. You have combinations of monocultures, same trees in the square. And maybe you don't see it really well here, but each of these squares you could see from Google Maps. So it was really defined. And you had 54 combinations that went from 1 species to 12 different species. And the whole design was replicated 4 times in 4 blocks. So really cool experiment that I didn't set up. So the team set it up. I just enjoyed sampling it, which was amazing. And these are the 2 gradients. So we asked the question, what influences tree community productivity or community productivity? Is it just the number of species? So the more species you have in your community, the better you'll be at harvesting resources? Or is it the functional diversity? That you will look different. And if you look different, then you probably use different resources. You use different layers in the canopy, you use different layers in the sun, in the soil, sorry. So for example, here you could have a balsam fir and a spruce and 2 species combination. So that would increase the species richness, but not really the functional diversity because 2 conifers look alike in terms of functional traits. But if you go from a monoculture to a 2 level of functional diversity, then maybe you have a balsam fir with a sugar maple, and then you have very different functional traits. They don't look alike, they don't use the same layers in the canopy, so they probably harvest resources differently. It's the same if you think of a football team that goes to a buffet. If everybody eats pizza, there's no more pizza 5 minutes in, but there's lots of other resources. But if everybody eats different stuff, then you can produce more biomass because everybody has more resources to eat. So it's the same for the hypothesis of the community diversity to productivity relationship. And these models are often monotrophic. We'll say we need to plant more diverse plant communities because that yields more biomass for the community. We think at the monotrophic level, plant only, but that probably has many different layers. If you have more diverse plant species, you probably have more diverse bacterial species as well, fungal species, insects, or other mammals maybe. So if you think of the ecosystem, this has cascading impacts. So for my second project of my PhD, we asked a question. Yes, we want to know if species richness, functional diversity, and functional identity at the plant level influences community productivity at the plant level. But we also want to know what happens to microbial diversity when you have more plant diversity. Does it mean that you have more diversity of bacteria when you have more diverse plant community? So we sampled the design and we measured productivity and we built this structural equation model, which is a path analysis, which is just many linear models together. Everybody good? So a path, yes, question.
What does BEF stand for?
Can you speak louder?
What does BEF stand for?
Excellent question. Biodiversity ecosystem functioning. So this is a field of research in plant ecology where biodiversity is related to ecosystem functioning. It can be resilience, it can be productivity, but you have this positive relationship between biodiversity and ecosystem functioning. One could say it's mostly studied in coral or marine environment and plant, but it's also been found in other environments now. And it's also debated as anything in science. You know, you will find recent papers that say maybe it's not always the case that biodiversity is related positively to ecosystem functioning. So thank you for the question. So with this model, what we could test is the direct impact of variables that were at the plant level on productivity, but we could also follow their impact through leaf bacterial diversity on productivity. So our model recapitulated what was already known. If you increase the number of species of trees in a community, you produce more wood. So this is what you see with the thickness of the arrows here. More species richness, more productivity. Functional diversity, more productivity. And functional identity was just a sense for us to be able to see which community produced the more biomass. So it's negative in the arrow, but it's just an ordination axis. So it doesn't mean that a specific functional diversity was related to negatively to productivity. It just meant that conifers actually produce more biomass than deciduous trees, or quicker. What was interesting to us is that even when we considered and factored in these very significant relationships in our model, there was still a significant impact of leaf bacterial diversity on productivity. And one thing I will say about this is this structural equation model is a hypothesis. This is not testing causality. I didn't demonstrate that increasing leaf bacterial diversity increases biodiversity productivity. But what I say here is that there seems to be a correlation between both. That when you increase the diversity of plants, this relates to a higher diversity on the leaf of your plants as well. And this correlates with a higher productivity. And this model explained 85% of the variation in productivity, which is pretty astounding for an experiment that is outside on the field. So this was interesting because it went into Nature. It was a very key piece in my career, and it was reviewed by 6 different reviewers. So it was quite something. Wow. And one said, well, actually, couldn't it be the opposite? Couldn't higher productivity of your community increase the diversity of the bacteria on the leaves? Couldn't it be the opposite? And we couldn't say it wasn't that, but what we could do is rely on other evidence from literature that had shown no relationship between increasing productivity of a host to increasing the diversity of its leaf, but the other way around had been true. So it seems that when you saturate the leaf of a tree, you can increase its resistance to pathogens because the niche is occupied. So it's harder for a pathogen to arrive. It seems that when you increase the diversity on the leaf, you can also have a better access to nitrogen for the plants because you have a greater chance to having nitrogen fixing bacteria. So there were a few hints, even at influencing phytohormones production, that could help us support this hypothesis. But this fat analysis is not a causality demonstration. So remember that. It's a nice statistical test, but it proposes a hypothesis. And in the lab now, we're trying to reproduce that with Arabidopsis, but to demonstrate the causal mechanism rather than testing the correlation. Perfect. So I finished this project, and I really liked trying to bridge the how the influence of the environment and the bacteria in the environment, the microbes in the environment, links to our own health. And when I was starting my PhD, Hansky had published this paper where the diversity in your environment influences the diversity of bacteria on your skin, and this could be related to allergy risks. And this was all correlation, but it was really interesting to start thinking about how what's in the environment influences our own health and how we manipulate the environment and thus have an impact on our health. So I think this is why when coming here and browsing the website of the One Health Microbiome Center, I realized I was maybe a One Health researcher and didn't even know it. I just never branded myself like that, but I find it very interesting. So this project was really to start to look, well, I've been in the natural forest looking at what is on the leaf. I've been in an experiment looking at what is on the leaf. What's happening in cities? Do we find the same things, or does it differ? So we designed this project where we looked at trees across the island of Montreal. So again, this is Quebec, all of this, nobody lives, or not many people live there. We're all right here in the south. And this is the island of Montreal. Yes, Montreal is an island. And most people live here downtown, but the west of the island is also field and more rural. And we designed a gradient where we would sample 7 tree species to look at what was present on the leaves and to compare it with what we had found in the natural environment. Again, we saw that there was a shift from the blue, which is the low urban gradient, to the red, the high urban gradient. Host species identity explain most of the variance in community, urban density, and we also look at tree isolation. So often trees are planted alone on the side of trees, on the side of streets, but sometimes they can be in parts. So we had dichotomized our data set that way. And with this model, we explained 57% of the variation in community assembly. So that was interesting. But when looking at the data, we realized that for some species, it really didn't matter where they were planted. They assemble the same community. For example, white spruce, I've highlighted just its dots across the gradient, and you see very little variation, at least on the 2 first axes. But when you look at red maple, it's a completely different story. You see a shift from the squares, which were the low gradient, to the triangles, which were in the high gradient. And so some species seem to have a higher sensitivity to conditions in the urban environment. So this finishes the urban section, but we'll come back to it because I've started new project in the topic. last of this section is sugar maple. So if you know about Canada, you probably heard about maple syrup. It's very important to us. Some say we drink it just like in mouthful, but it's not true for everybody. But it's a very iconic species, especially at this time of the year when it becomes red. It's so pretty, and we love to go hike in the woods, and it's a very important species. But it's not only in Canada. You see that in the US, it's also very present. And so one question that we have now is, with the increase in temperature, what's going to happen with sugar maple? Not necessarily for sugar maple syrup production, but more for the species per se. Is it going to be able to move northward? Because it seems that its fundamental niche might extend there. But if it can't, why is it restrained? So I was asking this question: is it going to retract? Is it going to march? This is one of the projects I'm most working on now. And I have a colleague in Sherbrooke, which is one of my idols. And it's really funny to work with somebody that you esteem so much, fangirling the person so much. Mark Velen is a great researcher and taught a lot about ecology and ecology of communities. And he did this experimental transplant at Mont-Mégantic, where he took some seedlings of sugar maple and transplanted them beyond their actual range to see what would happen to them. And what he found, and he's not a pathologist, he's not a fungal person, but he found that his seedlings that were at the edge of the distribution and beyond the edge of the distribution had more fungal pathogens on them. And he presented the results. He didn't follow through after that. He just showed that in his article. And I was like, but why? What's happening? Are there more fungal pathogens beyond the limit of sugar maple? Is it just the loss of its symbionts and then it's more vulnerable? Is it pH or something else in the soil? Or is it bacterial communities that are not the same? And this is what drives the change. Yes.
Just real quick to clarify. So is this a greater community of pathogens or greater disease?
It was pot with fungal infection.
Infection. Okay.
Yes, just visual presence of infections in the pot.
Gotcha. Okay.
That's it. So not a fungal pathologist, just saw that and reported it, which I found very interesting. So I designed this project where we went and sampled and just looked at what was happening in the rhizosphere and the phylosphere of sugar maple at the same gradient. And of course, we couldn't sample beyond the gradient because there was no seedling to find. But we ended at the edge and we looked at the phylosphere and the rhizosphere. And what we found was that for the phylosphere, it really didn't change much in terms of diversity. But in the rhizosphere, at the edge, which is the pale gray, you saw a shift or a loss of diversity. And which was marginally significant for the rhizosphere, which would be the ectophytes of the roots, but which was significant for the endophytes in the roots. And diversity is like this black box. There's a shift in diversity. What does it mean? We don't really know. So it was a hint that something beyond just increase in infection was happening. This was a master project. So we designed a second project where we would transplant these seedlings across a latitudinal gradient, across the temperate forest to the mixed forest to the boreal forest, and look at what community would assemble in the soil. And what we found, we looked at leaf bacteria, leaf fungi, rhizosphere, including mycorrhizae this time, because sugar maple is an arbuscular mycorrhizae species. So we wanted to have mycorrhizae on their side, and we included them. And what we found was in the boreal forest, which is all the greens here, for mycorrhizae, you see a significant loss of colonization, of richness and diversity in the boreal forest. So fungal infection on the rise, bacterial richness seems to be decreased at the edge. Seems like mycorrhizae might also be reduced in terms of diversity. But these were all observational studies with some experiment, but very limited in terms of output. So first project I did when I arrived in Sherbrooke was to find a student that was interested into microscopy. So we could look at the roots to have a visual of the colonization by mycorrhizae. And we designed this project where we had two gradients in Sutton and Mégantic. This is Vermont, this is Maine. And we sampled 50 seedlings at both locations. And we looked at the mycorrhizae by microscopy, the fungi, the bacteria. And we looked at the growth of the seedlings, the foliar nutrients, the soil composition, the plant community composition at the site. This was a master project that could have been a PhD. It was just so dense in data. But So we're just trying to get it published now. So I'm going to show you a few preliminary results. And one thing that struck us was that we would think that the growth of the seedling would be lesser or not so good at the limit of the sugar maple, because that would be where it's not really present anymore. But it was actually the opposite at both sides. The higher we would go in the elevation, the better the trees seemed to grow. And it wasn't due to better access to light. We controlled for that. It wasn't due to better access to water. Something was happening here that we didn't really understand. We looked at the colonization of the roots in terms of mycorrhizae, and physical colonization went down in one side, but not in the other one, at least not significantly. But we had some dark-septed endophytes that seems to be like funky guys that we don't really know what they do. They seem to be ambivalent of what they want to do, but they seem to play a role. So we might follow up on that route, but they were in lower abundance at higher elevation. So they might play a role in terms of influencing the growth. So we built again these structural equation model, and this is very rough. I'm sorry for it being very dense because it hasn't been polished yet, but this is for one site, a model where we ask what were the drivers of seedling annual growth. And what you see here is two compartments or two microbial community that drove increase or the growth of the seedlings. And those were root AMF, so root mycorrhizae, and fungi, composition of the fungal community. So they seem to be or two culprits in influencing the growth of our seedlings. But another one that we couldn't take out was just pure elevation. So it seems that even when we took into account and measured all these factors about the soil, about the plant community, there's still something that is happening along the elevation that we didn't capture. A shift either in community composition, distance to conspecific species. There's a factor that is changing along the elevation that we cannot explain by those factors that we measured. But we are onto something in terms of both distance to conspecific, which would mean that there might be a negative density dependent pattern here, that where you're closer to your conspecific, you're challenged by using the same resources. So we'll follow through this story, but otherwise soil pH was also very important, which kind of makes sense. Okay, so take home message for the plant section. How much time? 15. Oh, so I'll skip this one and I'll come back to it if you need a recap. But I wanted to talk about the human side. So let's use this time for that. So I went into my postdoc and I worked at the Germ-Free Microbiome Center in Calgary. They just opened it and it was really fun to work there. You had to take a shower to go in because you're full of microbes, scoop. And then you had to dress like an astronaut with like frocks and plastic bags that are awful for sweating and gloves and tape all over your body and caps and so on. And we would manipulate the mice. And if you forgot something, you had to go out, shower again, and come back in. So better not forget anything once you go in. But it was really amazing because when you think about trying to identify causal mechanism, this is really when you can actually put a specific microbe and see its influence on its host while acknowledging for as many factors as you can. So a privilege to go there, definitely a challenge as well, but really fun. So I'm going to talk a little bit more about the work I did there. So 2017, it's becoming very clear that microbes influence host health on a variety of aspects. It's influencing our immune system, it's influencing our metabolic system or metabolism, it's influencing how we metabolize medication. So we're discovering how many aspects of our life it influences. The part that I'm most interested into was how it would influence the immune system. And it relates to what I said about the urban chapter. How does the environment influences the training of our immune system? So I like that I could come into this thinking about not only the human microbiome per se, but how did the microbiome get into the human? So I arrived in the field of the human microbiome thinking about ecological dynamics. So how does infant get colonized by the mother, by the family unit, How do perturbations such as antibiotics influence the assembly of the microbiome in early life? And if you have or have had young children, they're sick all the time. And you cannot wait 1 month to see if they will survive without antibiotics. You kind of have to give them sometimes quick the antibiotics they need. My daughter had like in 1 year, 7 courses of antibiotics. And it's not that I'm against it. It's just I would rather avoid giving her too much. But it's just, it happens. And this has an impact on her microbiome. So I was really interested into this whole system from the ecological perspective. So the first thing I did as a postdoc is I wrote a review with Marie-Claire, and we looked at how colonization happened in the first 6 months of life. And so fungi are present in the gut very early in life. So there's a microbiome in the gut, and I'm pro Emily. And so what we saw was actually that the diversity of fungi early on is higher and then goes down, and the diversity of bacteria is the opposite. It's lower and then it goes up with exposure to the environment. And that was really interesting because it might not follow a linear path, because what happens when we give antibiotics, we reduce the load of bacteria in our system, and there might be a fungal bloom when we empty the niche of bacterial symbionts in our gut. So we designed a project where we would look at fungi, but we also had this partnership with a group in Xochicotla in Mexico, where they looked at protists in the human gut. Because there's not only fungi in the gut, there's also other members, protists that colonize our gut. And maybe in Canada and the US, sometimes they're associated with inflammation, but in countries like Mexico, sometimes they colonize up to 100% of the population without creating any inflammation. So this pathogen characteristic that we give in Western world and very developed communities might not extend to other countries. So we designed a project where we would look at blastocystis, which is a very common protist of the human gut. And we wanted to know what's its role for the community? How does it impact the other bacterial species and fungi? So we proposed this hypothesis that blastocystis is a predator in this environment. It feeds on bacteria in a fashion of winner take all. So the most prevalent bacteria will be the most frequently eaten. Not a preference for a specific bacteria, just I'm gonna eat whatever is in front of me. And this would create for a higher, more diverse community, because then it would reduce the domination or the dominance by one single bacterial taxa. So we had this project with blastocystis, and we wondered, is it a commensal, is it a pathogen? What's its impact on the bacterial community of the gut in the longitudinal cohort? And what we saw was that it's influenced, if I can skip the...
You might have to get closer. Yeah, there you go.
There. So what we looked at was the relative abundance of the most dominant taxa in the gut of our population. And we said that blastocystis, in the presence of blastocystis, which is in blue, we had a reduction of the relative abundance of Prevotella. And Prevotella is a fermenter, a fiber fermenter. It reduces the fibers and it transforms them in short-chain fatty acids. And it's really dominant in the human gut of our population. So presence of blastocystis did seem to have an impact on the community composition, but mostly on specific taxa that were reduced. We saw an opposite relationship as well on other taxa that were increased in the presence of blastocystis. We don't know if it's just because it's fed on the other taxa that increase the relative abundance of the others. And we look at the metabolites. So we look at the short-chain fatty acids, which if you've studied a little bit the human microbiome, you know that they have been associated with human health properties. And we looked mostly at acetate, butyrate, and propionate. And what we saw was a trend of decrease in the sample that had blastocystis, but it was really minimal. But it was in line with the reduction of Prevotella, which would produce these short-chain fatty acids. So less Prevotella, less short-chain fatty acids, at least a tendency in the human gut. So from this, we proposed that blastocystis might be an ecosystem engineer. And this goes to, if we go back to the microbe interactions, we have to think about these ecosystems as not monokingdom. It's not only about bacteria, it's not only about fungi. They interact together, they create this complex network. So what is blastocystis for us? Is it controlling the community from a top-down perspective in ecology, or is it more a bottom-up system where it's really the bacteria that control for the presence of blastocystis and maybe even its impact on the host? And when we take it out of or the common ecosystem and bring it by fancy trip to Mexico and it arrives in our gut, maybe it's not, our gut is not ready for it. But when you grow up with it, then you don't develop inflammation. So these are the kind of aspects of research that are still need to be answered. So there's lots still to do in the human microbiome. So colonization of germ-free animals. And I'm very sorry, I'm taking you on a very big loop. So I hope that you will get information out of this presentation. So this is a germ-free system, similar to Arabidopsis, where you could control its microbiota. So we colonized germ-free mice, so mice that were devoid totally of microorganisms, with either only bacteria, only yeast, or both together. And we looked at many different aspects, including the physiology of the animal, but also immune panels to see what was the influence of the colonization by these microbes on the host. And so you see first the cecum of these animals, then it's their GI tract, and this is the germ-free, and you can see how bigger it was compared to the three others. So this is germ-free animals, don't have any microbes, really struggle to absorb nutrients because they don't have their partner with whom they've co-evolved to absorb these nutrients. So what do? They produce bigger or more surface of their absorption organ to be able to at least get some of it. And so you see it here in intestinal length and cecum weight. So the yellow is the germ-free. And what was interesting was that when we colonized only with yeast, it was really close to what was found in germ-free animals. So recolonizing with yeast only is not sufficient to elicit a wild type-like phenotype. You really need bacteria to then get at a wild type-like phenotype. So bacteria is really needed. And what was most interesting was when we started to look at other aspects of the immune system. And when, for example, the fungi, the yeast-only treatment, looked again a lot like the germ-free, which have a very weak immune system and underdeveloped immune system. So we looked at a panel of immune cells with FACS, and we associated by correlation, again, it's not causality, many taxa, Candida albicans, Candida glabrata, that were associated with an overstimulation of the immune system that was akin to what we find when we have development of allergies and asthma later in life. So in this paper, they really demonstrated a causal impact of the yeast-only treatment on the overstimulation of the immune system, potentially leading to autoimmune diseases like asthma and allergies. Okay, so what about time now?
Pretty close.
Okay, 4 minutes. So we'll skip this one, but now you can ask me a question about it. We looked at the artificial sweeteners in the gut of infants and how it influences the human microbiome. And mostly what we wanted to know is, does mother that have a high consumption of artificial sweetener, how does it influence the development of the microbiome in the infant? The influence on the metabolic system and the increase in weight, so the BMI of the infant. And we found that 2 metabolites were elicited by the presence of artificial sweeteners, so aspartame, some of these non-nutritive that are 0 calorie for us, but not for our gut microbiota. So it was actually interesting to find metabolites that were associated with increased BMI at 3 months old and 1 year old. And that the influence during gestation of consumption of nutritional sweetener could have a legacy impact going up to 1 year after birth. This is a human cohort. So there's many things that you don't control. We did measure the type of nutrition that these people had, but you don't control what they get and what they report. So there might be confounding effects here, but what was interesting was that it really recapitulated what had been found in mice system, which are also limited because of what you can extrapolate from mice to human. So I'm gonna skip this take home just to talk about this final project, which is really one health centric. So the last thing I want to talk about is this project, People and Trees, a shared microbiome in an urban world. And during the pandemic, people were very limited in how they could move around. So this limited how they had access to nature. And we know nature has a great impact on our human health, it relaxes us, it reduces heart rate, but it also could have an impact on the development of the immune system in early life. So we designed this project to look at what's in the air in terms of contaminants, pollen, microbes, and how does it change across a gradient of economic status and access to vegetation. So this is very preliminary result that we got last week. So we sequenced pollen, tRNL, bacteria, fungi, and we have a contaminant measurement. And this is just the bacteria that we got last week in 3 cities that go from smaller to bigger. And you can see a clear shift between species, but most interestingly, you can see a clear shift in season. So we went and sampled every 2 weeks, and we really had a shift in the season of what you find in the air in terms of bacteria. So this is very preliminary, but we're looking on to have more data in the future. So with that, I will thank you for your attention, and I will take it.
One question from a friend.
I was just wondering if you could speak a little bit more about the dark septate fungi that you found. I study invasive species, and we're finding that it's associated with a lot of disturbance regimes. But like you said, it's really a mystery. So I was wondering if you could expand on how the presence of the dark septate fungi interacted with the other microbiome features that you found.
Yeah. Thank you for your question. A really great question. So we don't know much because this is a data set where we sequence AMF, a marker for AMF, a marker for ITS, and a marker for 16S. And we had microscopy of the roots of each of these seedlings. So we had visual detection, but we also had marker detection. We don't really know how it interacts because like we had snapshots, picture of the community. We know for sugar maple, it's not predictive, like it's not 100% response. So people have studied how it impacts sugar maple and Norway maple, and it's not coherent. So it seems to be either arbitrary or impacted by environmental factor or presence of other organism, but this is something I'd like to investigate more in the future. And the question I'm asking now is, how can I isolate them so I can test the causal mechanism of it?
I will be watching what you do to find out if you can do that. I study arid ecosystems, and they're a big factor in invasion for what I can see.
So I think there's something there, but we still are at the limit of, we know a few taxa that they represent, but there's more. And one thing and I was telling Seth about the assignation of taxonomy and function for fungi is just awful. It's just so limited. You think bacteria is limited, fungi is worse. It's really difficult, even just assigning with fungi, the expected function of fungi are just difficult to work with.
Well, let's make Isabelle.
Thank you.